Hello friends, this video on biomolecules part 24 is brought to you by examfear.com. No more fear from exam. Correct? So let's see the chemical composition of nucleic acids. Let's understand the nucleic acids more from the chemistry perspective, and here we will talk about the nucleotides also. So if we talk about the chemical composition, so when you do a complete hydrolysis of DNA or RNA, what you get is, just to understand the chemical composition, the best is you do a reverse engineering. So the chemists did the reverse engineering and they did the chemical complete hydrolysis of DNA and RNA, and they found that it yields pentose sugar, phosphoric acid, and some base, nitrogen containing base, right? So this was they got a phosphoric acid, they got two different kind of sugar, and they got some base. So if we talk about the nucleobase, in DNA we have four nucleobase: adenine, guanine, cytosine, and thymine. So these four are for DNA. In RNA, instead of thymine we have uracil. So this is for RNA and this is for DNA, and other are all common to all. So if you see, if you compare thymine and uracil, you see there's only one difference. Instead of H, there is a CH3 here. Everything is almost same, right? And that is why, because uracil, RNA has OH, ribose nucleic acid that has two OH, and it is not able to take this big compound, thymine, so it takes uracil, small difference. So that's the nucleobase for RNA and DNA. If you talk about nucleoside, nucleoside is nothing but nucleobase bound to ribose. Nothing but nucleobase, which we have just discussed, bound to a ribose or a deoxyribose. Deoxyribose, any of these. Right? Example, this is my ribose, this is deoxyribose. It is bound to all these base. So these are examples of output. Correct? This is my base. It can be a ribose or deoxyribose. So you get different kind of compounds based on whether you used which kind of base. You see, all these bases are different. Correct? So that is my nucleosides. Now on the nucleosides, if you add a phosphoric acid, you take a nucleoside and you add phosphoric acid, you get nucleotide. For example, this, if you see, this is a phosphoric acid added. So this whole thing is a nucleotide. This is the case of ribose nucleic acid, and this is the case where we took deoxyribonucleic acid. There's a base here. If you see, these bases are all these bases here, 5 bases. Correct? So from base you make nucleoside, from nucleoside you make nucleotides, and my nucleic acid is nothing but a long chain of nucleotides. That's also called polynucleotides. Correct? So these nucleotides are building block of nucleic acid, both DNA and RNA. So if you see, it has nothing but a sugar ring, a nucleobase, and a phosphorus group. And these are the base, the main building block of nucleic acid. So now we have got a lot of nucleotides, right? Now we have to form a chain of nucleotides. So there has to be a bond which connects this nucleotide. So we'll talk about the nucleotides bond. So all these nucleotides are joined together by phosphodiester linkage. So you'll see this. This is my phosphodiester linkage between carbon 5 and carbon 3. So this, we see, this is my nucleotide. This is my nucleotide. It has a base, sugar, and phosphor. Base, sugar, and phosphorus. So this are my two nucleotides. They are joined by phosphodiester linkage, carbon 5 and carbon 3. But this is carbon 5 and this is carbon 3. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. This is my carbon 5 and this is my carbon 3. So this is the link between these two. Correct? Actually, the link is not between these. The link is between this. Correct? See, OH minus will leave, it will develop positive charge and this will bond. That's what is happening. So they'll form a bond. This is called phosphodiester linkage. Correct? That is my nucleotide. And several of these nucleotides bond, if you see, they'll form a nucleic chain. Several of these will form a nucleic chain. So it will be sugar, base, phosphate. Sugar, base, phosphate. Sugar, base, phosphate. Something, right? Sugar, base, phosphate. Sugar, base, phosphate. Sugar, base, again, phosphate. Like that, the chain will continue. Correct? So, if you see, the nucleic acids also have structures, primary structure, secondary structure, right? If you see, this is my DNA, this is my DNA, this is my RNA. They have structures to understand it. So this is a complex structure to understand. So what chemists have done is this, they also subdivide this nucleic structure into. Thank you. Visit examfear.com to watch more videos, attempt free online tests, get free study materials, find tutors and mentors, and much more. Thanks once again.